Assalamualaikum and greetings. Welcome to our video presentation entitled Pinitol. For this assignment, the group members are Siti Ainsyah Binti Busu, Naakshectra anak perempuan Nadarajan, Vithyashini anak perempuan Sivanesan, Irfan Hakimi bin Bakrul Laini, Liong Xian, and Syahiera Erissa Binti Shahriza.
Pinitol is a pharmacologically active compound occurred naturally and generally belongs to the important family of inositol. The name originally from pine since it was isolated and identified from the hardwood of Pinus monticola pine as it was at first isolated from pine tree. Pinitol has pharmacological significance. It can also isolate from synthetic and semi-synthetic method by the means of various processes such as chemical and biochemical transformation. The major constituent of soybean plant is known as an insulin mimicker. There is a growing interest in the use of the Pinitol as a food supplement because of its reported efficacy in lowering blood glucose levels with no side effects.
Pinitol, also known as D-Pinitol, belongs to the class of organic compound which is cyclohexanol. Cyclohexanol contains a group of alcohol attached to its cyclohexane ring. The chemical formula of Pinitol is C7H14O6 that attributes to its molecular weight of 194.183 gram per mole. It has a high melting point of 183 degrees Celsius and boiling point of 250.62 degrees Celsius. The estimated density of Pinitol is 1.2501 gram per mole. Pinitol is a highly soluble cyclitol in water and slightly soluble in ethanol. It is the most dominant component of low molecular weight carbohydrate fraction of legumes such as carrots and soybeans. Pinitol is an extremely weak basic which is closely to neutral compound due to its high pKa value of 12.56. The Pinitol is a white-colored powder and it possesses multifunction properties such as anti-diabetic, anti-fibrosis, anti-hepatotoxic, antioxidant, anti-tumoral, and anti-inflammatory properties. The Pinitol is a methyl inositol extract that helps the transport of glucose and glycogen synthesis. Lastly, Pinitol promotes the absorption of creatine. Creatine is a well-known enhancer that is often used as an additive.
As the definition of building block is, a block is a time period in chemistry that's used as a digital molecular development of an actual chemical compound, the molecules of which on reactive cell groups. Building block involved for the Pinitol is 1,2-isopropylene dioxide, 3,4-epoxy, 5-cyclohexane. Synthesis of building block consists preparation of the NNTP of 1,2-isopropylene dioxide, 3,4-epoxy, 5-cyclohexane, involved by combining the enzymatic cycloaddition of isopropylene dioxide, cyclohexadiene, to chiral-chlorotrosol with an internal substitution of an amino alcohol to create vinyl epoxide. All method involve the use of protected vinyl epoxide to A and to B as key building block for the synthesis of Pinitol. For the biosynthesis of Pinitol is the D-Pinitol biosynthetic pathway proceeds from the precursor glucose 6-phosphate to D-Pinitol through a series of processes such as myo-inositol 1-phosphate synthase, inositol monophosphatase, and myo-inositol or methyltransferase.
So isolation of phytochemicals like Pinitol is a process done to remove constituents of plant extracts and purifying them into monomer compounds. And this is done by physical and chemical methods. So common isolation methods are actually divided to classical ones like solvent extraction, crystallization, and also modern separation techniques like column chromatography, high-performance liquid chromatography, and ultrafiltration. So usually what we do, we get powdered plant material. It could be any sort of plant species that contains Pinitol, and this powdered plant material at around 500 grams is kept in 95% of ethanol around 2 liters and also kept in a percolator for 15 hours at ambient temperature. The solvent is then drained off and repeated 4 times. So the reason why the solvent is repeated 4 times is to make sure to remove any sort of impurity that could actually be caused in the material. And then all these drain off portions will be combined, filtered, and evaporated under diminished pressure to yield residue. The residue is then triturated with hexane to remove non-polar parts of extract, and the residue is then going to be subjected to column chromatography. So you see, even before the column chromatography, a lot of steps are actually taken to remove any sort of impurity to make sure that we get a really pure yield at the end of the process. And the polarity of mobile phase will actually be increased by the addition of methanol gradually. So column chromatography is usually used in this kind of processes because it is a cheap process, a wide range of mobile phase can be used, and also it uses a robust metal.
Next is the biological activities and therapeutic values of Pinitol. Pinitol provides many benefits to the society. They are particularly important due to the diverse biological activities and also therapeutic values. Pinitol serves as a physiological cellular activator as well as a chemical defense in plants against unfavorable conditions. It is regulated from plants as a production of tolerance to environmental changes in response to osmotic stress conditions, including water scarcity and excessive salinity. There are few studies where pinitol was considered as a computable solute, in which they can act as an osmoprotectant to allow turgor pressure to be maintained at a lower water potential, as well as for osmotic compensation. This compound also can act as a chemical defense due to its synthesis in plants. It also can act as a cryoprotectant in cold stress. Additionally, insulin plays an important role in regulates the translocation of glucose transporter type 4, GLUT4, from the endoplasmic reticulum to the plasma membrane of skeletal muscle to maintain glucose homeostasis. Therefore, by using pinitol as the treatment, it can enhance the glucose absorption. In fact, there is more recent research reported that by using pinitol, it can increase the membrane translocation of GLUT4 in the skeletal muscle and decrease the plasmatic levels of glucose and insulin. In addition to its insulin activity, plants that rich in pinitol can possess many therapeutic values such as anti-diabetic, anti-inflammatory, antioxidant, anti-fibrosis, anti-cancer, and many more. It is widely used to possess multifunctional properties in traditional medicine as empirical treatment mainly for diabetes, infections, cancer, or inflammation conditions. Pinitol could be beneficial in the treatment of inflammatory illnesses such as hepatitis and also rheumatoid arthritis. In anti-cancer and chemoprevention, the mechanism of action of this medication as an antitumoral has produced mixed results. This can be seen when pinitol was able to increase apoptosis and suppress tumor necrosis factor (TNF), which induced cellular invasion when cells were treated into pinitol in combination with TNF. Moreover, anti-tumor properties of pinitol have also been investigated in breast cancer cells and also mammary gland rat model. Besides, this compound have received more attention among researchers in past few years due to its assistance in foods and medicinal plants, which are including several species from Rhythomagenous, soybean, and fenugreek seed, which can give benefit to the people that suffer from diabetes and other diseases. Additional research on this drug is needed to have a better understanding of the underlying mechanism of action and to truly confirm the potential medical applications in disease prevention and therapy.
For the dihydroxylation method, they'll have the Hakimi synthesis of plus pinitol and Hakimi synthesis of negative pinitols. And for the exposition method, they'll have the Li synthesis of pinitols. Now we go to the dihydroxylation method of Hakimi synthesis of negative pinitols. Hakimi and other else also reported the synthesis of the negative pinitols by taking advantage of the Western symmetry style structure settings. This initial matrix switch was achieved by changing the order of the reaction sequence. The enantiomer of the nitrous product was obtained after the stereoselective expositions of the acetonides protected the diol 13 before the removal of the bromine atoms exposite opening with the methanol, which is structure 25. Dihydroxylations of the remaining double bonds and the subsequent cleavage of the acetonides. The possibility to access both enantiomeric forms of the pinitols and the demonstrations of the strategies principles increases when the scientific value of the synthesis. For the Hakimi synthesis of plus pinitols, he also reported on the synthesis plus pinitols start from the bromodiol 13 structures. The bromodiol structures 13 has been transferred into the corresponding acetonides and the covers close chains into the subjected to the selective oscillations from the much less hindrance side phase, which is structures 19. Reductive eliminations of the bromide chains into the completed earlier than the expositions of the closing double bonds the usage of the M-choral peroxide benzoic acid, which is the MCPBA. The exposure of the exposite structures 21 to the methanols below the aluminium oxides catalyzed via the methyl ether structures 22. The synthesis of the plus pinitol, which is structures 23, changes into the finished with the edge of using the cleavage of the acetonides protective institutions below the acidic conditions. Next, we go to the exposition methods, Li synthesis of pinitols. In 1987, Li and others published the synthesis of the racemic pinitols and the first academic application of the cis-cyclohexane diol is outlined in the following sections discussing the exposition as a diol-stereoselective reaction state. The structure 15 were protected as the benzoate before the materials were subjected to the MCP-BN medium expositions. Before the structures 1969, the small amount of the distereomeric material structure 70 were formed, which will easily be removed by flash columns chromatography. Compared with the benzoate, the cyclic protecting groups of the diol moiety, such as the acetonide or other acetals, were better suited to the efficiently block one phase of the cyclohexane diols, as evidenced by the subsequent contributions of the researchers into this field. The methanolysis under the acidic conditions selective yield the structure 71, which was used in a dihydrosilation reactions with the osmium tetrasides. The structure 72 was obtained along with the derivative of the structure 73, with the benzoyl groups migrating to the previous unprotecting hydroxyl moieties. The hydrolysis under the basic conditions yield the natural products, with the utilization of the mesodiol structure 15 as the starting materials. Pinitol was obtained in the racemic form, which does not decrease the merits of this visionary's work. With the first preparations of the pinitols, the benefit of the combining the advantage of the chemical synthesis and biotechnologies become previous to the large audience.
So that's all from us. Thank you.